Pleasure for me to attend and present this talk at the I2K conference this year. I'm Anuradha Kar, a postdoctoral researcher working at the Paris Brain Institute, and today I will be presenting on methods for exploring the capabilities of deep learning pipelines for 3D segmentation of microscopy image datasets. So here is the overview of the talk. We will take a look at a few deep learning based pipelines which can be used for 3D segmentation of microscopy images. Here we are looking at instance segmentation, where each segmented object has its own label or identity. Then we will explore various ways in which we can analyze their performance levels. Finally, we will look at several open source tools which could be used for evaluation of 3D segmentation pipelines like the ones we see in this presentation. First, we need to know about the data we have used for training and testing the deep learning pipelines. So we have two datasets of 3D confocal microscopy images of the Arabidopsis shoot and floral meristems, which are multicellular tissues. For training the deep learning models, we have 88 stacks of shoot apical meristem, and for testing, we have 10 stacks of floral meristem data. Each of these 3D images have their expert ground truth segmentations. This is an example of how this data looks like. So the objective of all the segmentation pipelines that we will see today is to segment or extract the cell regions from the images as individual identifiable objects. Now we will take a look at the segmentation pipelines one by one. First, we have PlantSeg from the original paper published in 2020. It uses a deep learning model called 3D-UNit. This can be trained on microscopy images to extract cell boundaries as we see here. Then it uses a post-processing method called graph partitioning to get the final 3D instance segmentations. This is an end-to-end 3D pipeline, that is, it takes 3D input images and produces 3D segmented outputs. Next, we have another end-to-end 3D pipeline which also uses a 3D-UNit model to extract three classes of outputs from the confocal images, which are cell boundaries, background, and cell interiors. It uses a watershed-based post-processing to get the final 3D segmented outputs. Next is a hybrid pipeline, hybrid because it can be trained using 2D data to produce 3D outputs. In this case, a UNit deep learning model predicts vertical and horizontal spatial gradients and cell probabilities, which they use to finally identify cellular objects in the images. The fourth deep learning pipeline is also hybrid. It uses a masked CNN model to predict 2D segmentations of all regions, which are then used with a watershed-based post-processing to get the 3D segmentations. You will get further details about these pipelines in our recent publication, which I will talk about in the end. Also, beside the deep learning pipelines for comparison, we have a classical or non-deep learning pipeline, which is from this publication given here. It uses a 3D watershed and is named Mars. It does not need training data, but includes manual tuning of the H minima parameter it has for getting optimal segmentation results. For testing these 5 segmentation pipelines, as mentioned before, we have a test dataset. Now, within the test dataset, we have 10 original confocal images from different time points of development of the floral meristem. In addition, several microscopy image artifacts were also simulated on this dataset to test the impact on the performance of the 3D segmentation pipelines. These artifacts include blur, partial over and under exposure, and 2 different levels of Gaussian noise which are added to the images. So finally, we have a test datasets of 60 confocal stacks for testing of all the segmentation pipelines. In order to explore the performances of these 5 segmentation pipelines, we have set up a protocol as shown here. As said before, we have a test dataset which has original confocal images and also artifacts simulated on them. Next, we apply each of the 5 segmentation pipelines to segment this test dataset and then study the segmentation accuracy, compare the relative accuracy levels using different metrics and visualization strategies. To estimate the performance of the 5 segmentation pipelines, we use a 3D segmentation evaluation metric which is called the volume averaged Jaccard index, defined by the formula as shown here. It is an overlap-based metric like the Jaccard index, but is estimated on voxels for comparing 3D segmentations and is averaged over the volume of the ground truth cells. Using this metric on the results from the 5 pipelines, it is seen that the PluntSeg performs best out of the pipelines in terms of segmentation accuracy. The non-deep learning pipeline MAS performs equally well. Accuracies of the hybrid pipelines for 3D segmentation are lower than the end-to-end 3D pipelines. After testing the pipelines on the original confocal images from the test dataset, we now test the 5 pipelines on the confocal images where different artifacts have been introduced. So first, we have images where noise is introduced. Noise in microscopy images may be caused due to the electronic detector or amplifier of the imaging equipment. So here we simulate 2 different noise levels for our test stacks and use the 5 pipelines to segment the images and estimate their segmentation accuracy. The accuracy results are shown in this plot, where the first box shows accuracy of the pipelines on original data and the next 2 boxes are accuracy levels for Gaussian noise variances of 0.04 and 0.08. We can see from the results that out of all the pipelines, the PluntSeg is least affected by noise, whereas other pipelines show a sharp drop in segmentation accuracies in presence of noise. Next, we apply the 5 pipelines to test images where the blur effect is simulated. Image blur is often caused in microscopy images due to lens aberrations or motion effects. On testing the segmentation results on images with blur and estimating the accuracy levels, we found that only the 3D unit with watershed pipeline is considerably affected by this artifact, but the other pipelines not so much. Finally, we test the pipelines on the over and underexposure artifacts. Overexposure is caused due to factors like inhomogeneous illumination, while underexposure or shadow regions may be created by light absorbing objects or obstructive parts of a sample. From the accuracy results of the 5 pipelines for both under and overexposure, it is observed that PluntSeg and the 3D unit with watershed are seriously affected by these image intensity artifacts. The non-deep learning pipeline MAS, on the other hand, is affected much lesser compared to these 2 end-to-end 3D deep learning pipelines. Now that we saw that different deep learning pipelines behave differently in presence of different microscopy image artifacts, the question arises that what happens if we add one or more of these artifacts in the training set for the pipelines. This is what we tested here. For this, a set of stacks with the overexposure effect and a set of stacks with underexposure are used for training the PluntSeg and the 3D unit and watershed pipeline. So there are 3 experiments done for each pipeline. For example, in the first experiment, we retrained the PluntSeg pipeline with normal and overexposed data. In the second experiment, with normal and underexposed data. And in the third, with normal, over and underexposed data. The same is done with the other deep learning pipeline. From the results, as we see in the plots here, the PluntSeg pipeline shows definite improvement on adding microscopy images with artifacts in the training set, especially when the images with shadow are added to the training, or all the data are mixed. Whereas, if you see the plot for the 3D unit with watershed pipeline, you will notice that this is not the case. Its performance actually degrades with the addition of under or overexposed data in the training set. So the takeaway knowledge from this is that not all 3D segmentation pipelines using deep learning will respond in the same way if you add data with artifacts in their training set. It may make some pipelines robust, but it may not do so for the other pipelines. So far, we have been using the volume average Jaccard index metric. Now we will talk about a metric which classifies segmentation errors. So the VGI metric tells us that the magnitude of errors that we could compute, but there are different types of segmentation errors. These are errors due to over-segmentation and errors due to under-segmentation. In over-segmentation, an object in the image is split into multiple regions in the segmentation. In case of under-segmentation, the opposite happens. That is, multiple segmented regions fuse together to form a large region. We adopt a metric here which can differentiate between these 2 classes of errors, and you can actually compute the percentage of errors in a predicted segmentation into these 2 classes of errors. This metric takes as input a segmented output from any pipeline and the corresponding ground truth stack. It then provides the number of occurrences for each class of segmentation errors present in the given segmentation. That is, how many cells have perfect segmentations, how many are over segmented, and how many are under segmented. Using this metric, we can again look at evaluating the 5 segmentation pipelines we have. So, the rate of over segmentation or the number of over segmentations divided by total number of cells in the images is estimated for all the 5 pipelines and is shown in the plot here. We see that for all the pipelines, the rates of over segmentations are below 20% except for the masker CNN pipeline. When we estimate and plot the rates of under segmentation, we see that for plant-seg the rates are still low while it increases for the 3D unit with watershed pipeline. Now, you can find further details about this metric in our paper and open source implementation in our repository that I will be presenting at the end. While metrics provide us a quantitative idea about the segmentation performances of the pipelines, it is also important to be able to visually observe where the errors are within a segmented image. For this, we developed a 3D visualization method using Morphonet, which is a browser-based 3D morphological data visualization platform. Through this platform, we can upload the 3D mesh obtained from a 3D segmentation and also upload segmentation metric results and superpose them on the mesh using color maps. This can help us in visually inspecting which parts of a segmentation have high and low accuracies, even going to the cellular level. Here is a demo of how we can visualize segmentation accuracy results in 3D using Morphonet. We have uploaded a 3D mesh obtained from the 3D segmentation of one of the confocal images from our test dataset. This is what an uploaded mesh looks on Morphonet. Now, in the drop-down menu, we can see that all the accuracy metric results from all the pipelines have been uploaded on the Morphonet platform. Now, we can select one of the pipelines and superpose its metric results on the 3D mesh as a color map as we see here. Using the Morphonet platform features, we can now rotate the mesh in 3D, slice through it in XYZ directions, or even view the segmentation metric result for each cell by clicking on it. This visualization method is completely open source and the full implementation details are provided in our paper and repository. And this is what segmentation results from each pipeline looks like on Morphonet for a segmented stack. The red colored cells represent where the Jacquard index is high and blue cells where Jacquard index is very low. So we can directly see here that for this particular segmented stack, the plant-seg and Mars has very few errors, but the other pipelines, there are many regions which have low Jacquard index. So we come to the conclusion of this presentation. We explored the performances of 5 segmentation pipelines. 4 of them are deep learning based and 1 is a classical 3D watershed method. We saw that both the deep learning and non-deep learning 3D segmentation pipelines have comparable performances. For the non-deep learning methods, the main issue is that it is time and effort consuming to get the best results because these methods require manual parameter tuning. The deep learning pipelines after training need minimum human supervision, but they need a lot of images and annotated ground truth data for their training. The takeaway from this talk is that open source metrics and visualizations are very important for the scientific community working in bioimage segmentation because this helps to gain deep insight into the strengths and weaknesses of the many segmentation pipelines that currently exist. Therefore, all the codes and data used in this work are open source and I will present them to you now. First, this is our repository named segcompare whose link I have included here. You can scan the QR code to get the link to it. In this repository, we present all the coding implementations of the metrics and visualizations that I showed you today. There are also Python environment setup files which will help you to run these codes in a very easy way. Then there are training and test data sources and sample files and demo videos which will also help you to use these resources. And this is our publication which presents all the 5 pipelines describing the benchmarking protocol to evaluate and compare them and thorough details about training and using these segmentation pipelines along with details on the metrics and visualizations. It is published in PLOS Computational Biology in April this year. Here are my acknowledgements. Finally, I just wanted to let you all know about a new open journal called Applied Biosciences, which started this year, and I am in the topical advisory panel for it. It covers a lot of topics in bioinformatics and computational biology, so may be of interest to you. And so thanks a lot for listening to the presentation. If you have any queries, you can contact me by mail given here.